this is our video to help us practice what we just learned. And you'll be happy to know this is take 3. I had a little bit of problems with the other ones, so hopefully I got it. So if you open up your biology cell respiration jam, you're going to see slide 1, which is just to help you practice locating and finding everything. So notice we've got in our yellow sticky notes our 4 parts of the mitochondria that we're going to be looking at a little bit more. And then we can even add one here for the cytoplasm. And just to really clear that up, the cytoplasm is going to be outside there we go. So that's going to be our cytoplasm there. Now I'm going to go ahead and make a copy real quickly because I need to make a key. But as you guys are seeing this, you guys, while I do this, you can go ahead and you're going to move your sticky notes to the right part of the mitochondria, and then you're going to match the green sticky note to the right yellow sticky note. Where do they occur? So go ahead and take a minute. You can pause the video and see if you can figure out where everything goes. So let's see if you got it right. So here's our cytoplasm, and glycolysis got the wrong thing. Glycolysis is going to be in the cytoplasm, so we can match those 2 up. And then let's see what else we've got. We've got our outer membrane, the dark brown, and then we got our inner membrane, our lighter brown. And then the matrix is going to be the inner part of the mitochondria, the blue. And then we got this space. Now, we haven't talked about that intermembrane space yet, but that is going to be coming up. We'll see. And hopefully you found that the Krebs is in the matrix and the electron transport system is in the inner membrane. So that is what you should know. So for slide 2, there's not much you need to do for this one. I just thought it would be kind of nice to show things in a more visual way, what the goal is. So remember, let me use my little laser here. Remember the goal. Here's our glucose with all of those wonderful high energy electrons that are trapped in those bonds and hydrogens that are accompanying those high energy electrons. And remember, what I told you is watch for those falling electrons, like watch where they go. And so I had a little fun here with my little construction workers. And you can imagine them saying, wow, look at all those electrons and hydrogens. And the other construction worker says, let's remove them, which is what we're going to do. And so what you see here is glycolysis, Krebs cycle, and electron transport chain. And I didn't do all of the products, but what I wanted to focus on were those energy carriers. Where is the energy going? So, and sometimes when you watch the crime shows or FBI shows or detective shows, they always say, follow the money. Well, let's follow these electrons. Let's follow what's going to happen to them. So in glycolysis, notice what we're going to do is we're going to start the process. Here's our NADH. And remember, we make 2 of them, and then we get 2 ATP, net, 2 NADH, and 2 net ATP. So we're stripping some of them off, and we get pyruvate, and then we're going to keep going with the conversion, and then to the Krebs cycle. And in the Krebs cycle, we're making this huge convoy of NADH. So you can see if I did it right, there should be 8. 1, 2, 3, 4, 5, 6, 7, 8. And then 2 FADH2s are little bulldozers, and then 2 ATP. So we don't really have that many ATPs so far. We got 2 in glycolysis, 2 in the Krebs cycle. But notice somewhere between the Krebs cycle and the electron transport chain, we end up making a bunch of ATP. And the big question that we kind of have here is, how do we do this? We've got a lot of ATP coming, But where did they come from? Well, that's going to be our next stage, our last stage. Now, I just wanted to do a quick little schematic for glycolysis, again, just to practice. So notice here are my little sticky notes for the goals. Use the energy in glucose to make 4 ATP, but we need to add ATP to boost it up. So let's start with glucose and do your best with your arrows. You can always add arrows in the shapes. I'm going to use my highlighter. Now, the first thing we can do with glucose, and this takes actually 4 or 5 steps, but we're going to take glucose and we're going to split it apart. And notice we split it apart into 2 3-carbon molecules. Now, the way we do that is we need to add 2 ATPs. So I'm gonna add an ATP. This is the energy investment stage. And you can use a text box for this, or you can write this on a piece of paper, either one. But you do want to have it on the Jamboard at some point in time. So we're going to add ATP to this process. And by doing so, we're going to break it apart. We're going to get ADP and a phosphate. Again, you can do a text box. Now, we're going to do this twice. So I'm going to go ahead and add a 2 here. And we're going to add energy to this glucose just to bump it up and get it started, just like pushing a boulder down the hill. So we're going to do that. We're going to start the process. We're going to split it up. And now this is going to happen on both sides here. We're going to be able to, in the process of getting our pyruvates, we can get 2 ATP out. Oops. 2 ATP out on both sides. And this is where our net comes in. We had to notice the arrow. We had to add 2 ATP. And now we get 4 ATP out. And so there's where our 2 net ATP comes from. And then we're almost done. We're going to be able to pull out 2 NADH. One on this side. And then one on this side. And that's what you should have. And notice we're left with 2 pyruvates, 2 NADH, and 4 total ATP. But remember, we had to put 2 in. And so that was our little practice for our glycolysis. All right, so that should hopefully give you some practice.